Hi, I'm Jason. I'm a teacher, so I don't make much money. This spring, I needed a bunch of trees to plant, but I didn't want to pay for them. So here's a story about trying to figure out what makes cell division happen so it could get the trees to grow themselves for me.
Most of my property used to be cow pasture, and that's why there's really not a lot left out there as far as cover for the wildlife. It's winter now, but this spring I want to do a bunch of habitat restoration on it by planting a bunch of trees. Unfortunately, buying all those trees can be very expensive, so I want to try to find a way to just increase what I already have out there to provide more cover for the wildlife. So what I would really like is to get more of this plant growing out here. It's a shrub called red osier dogwood, and it's great for wildlife. The birds love it for nesting and for the berries it produces, and the deer love it as well. These red twigs are really nutritious, and in the winter they provide good browse. You can buy these dogwood seedlings at a nursery, but they're going to run 10 to 20 bucks a piece. And for the area I have to cover, that would be way too expensive. So our experiment is going to be trying to figure out a way to multiply what we've got already. So when I was researching habitat restoration, I read some information that certain species like dogwoods and willows can start an entire new plant just from a stem cutting from an already growing plant. So apparently if you stick it in the ground, new roots will develop and it'll grow into a new plant. And that would be great because I could do this entire area without spending any money at all. Unfortunately, the techniques didn't seem like they were 100%. So before I spend days and days doing the planting in the spring, we're going to take these inside and do some experiments to find the most effective way to get these to start new roots and grow into a new plant. For this to work, we're hoping that this process happens, cell division and differentiation. We want the plant cells down in our bottom of our cutting here to divide into new cells, and then when they divide, differentiate into root tissues instead of stem tissues. And that can work because every single cell in the plant has the genes for any type of cell. We just need the right genes to get expressed. And the process that's going to make that happen is by giving these some sort of internal or external cue that's going to trigger them to release hormones, and those hormones are going to tell the cells to divide and then differentiate into root tissues. We'll run 5 different treatments to try to increase the root development on these shoots. The first treatment is just going to be the control, and we'll stick the cuttings in a mixture of sphagnum moss and sand, and hopefully within a few weeks there'll be roots forming on them. For the second treatment, we're going to try to throw an external cue at the plant cells to make them think it's spring by soaking them in water. So 24 hours in there, and then I'll put them in a bucket just like the control. For the next treatment, we're going to try to bypass the process of those internal or external cues and just put hormones directly on the shoots. And we can do that because they sell rooting hormone at the nursery. And what we're going to do is just sprinkle it right on these shoots and see if it'll trigger them to divide and become roots. So we put the rooting hormone directly on the shoots. Now we're going to combine it with the water and see if the combination of soaking plus the rooting hormone has a better effect. Our last treatment goes along the theme of not spending a bunch of money on this restoration. So while I was doing research on how to get these to root, I found a suggestion that you put them with willow cuttings because the willow cuttings produce a bunch of hormones that can then affect the dogwood cuttings. And we're just going to put them in the water with the dogwoods for the 24 hours before we plant them.
Let's review what we did. We put a bunch of tree stem cuttings in different conditions to try to get their cells to divide rapidly and grow new roots. And after a few weeks, it was time to check them and see if we had any success.
On the surface, it looks like everything is going really well because we've got leaves growing from the cuttings. That means cell division did occur to make the leaves, but we want to make sure it occurred below ground as well to make the roots. I'm going to carefully dump out each bucket into this container to get the shoots separated from the dirt, and then I'll rinse them in this bucket of water so we get a clear view of any roots that are growing from the stems. I'm definitely seeing some roots on the part of these cuttings that were under the soil, but I'm going to look through them a little bit closer and try to come up with a ranking for which ones worked best and which ones didn't work so well. It looks like last place is going to go to the cuttings that were soaked in water for 24 hours. Those had the lowest amount of root growth on them. The definite winners were the willow cuttings and the rooting powders. Those worked because they sent some chemical signals to the cells of these shoots to tell the cells to divide and to tell them to differentiate into root cells. And they grew really fast. If we looked at these under the microscope, we would see lots of cells in the phases of mitosis, like prophase, metaphase, anaphase, and telophase, because they're rapidly dividing down here in these new roots that are growing. Even though it was the most expensive option, it was definitely the most effective, so I'm going to use the rooting powder and water treatment on all of the cuttings when I do my habitat restoration this spring.
That was an example of using a little science to solve a problem. I was able to save a bunch of money and improve my property just by putting a bunch of sticks and dirt to make cell division happen.
Bacterial cells increase in numbers by the process of binary fission. The parent cell grows in size while doubling all of its contents. Then it divides to become 2 daughter cells. Each daughter cell is identical to the parent cell. 2 cells become 4, which become 8, and so on. This results in exponential growth of the population. The time required for the number of cells in the population to double is called the generation time. Depending on the medium, temperature, and other conditions, some bacteria can double in minutes. Other bacteria have generation times of several hours or days.
The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or GAP1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or GAP2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers, that can be used to form the cytoskeleton of the daughter cells.
During interphase of the cell cycle, the genetic material of the cell is found in the form of chromatin and located within the nucleus of the cell, which is surrounded by the nuclear envelope. During the S stage of interphase, DNA replication takes place, and thus the cell leaving interphase and entering mitosis already has replicated its genetic information. In addition to the replicated genetic information, interphase cells that are about to divide also replicate their centrosomes. In animal cells, these centrosomes are formed from centriole pairs. As cells leave interphase and begin prophase, the first stage of mitosis, the chromosomes begin to condense. The nuclear envelope begins to vesiculate, and the centrosomes migrate to opposite ends of the cells to define the two poles. During prophase, the mitotic spindle apparatus begins to form by the polymerization of tubulin proteins. This creates microtubules that emanate from each centrosome. Chromosomes completely condense such that sister chromatids formed from the replicated genetic information and joined at their centromeres are clearly visible. During prometaphase, the nuclear envelope completely breaks down and the mitotic spindle is formed. Polar microtubules extend from both poles and overlap with each other. The sister chromatids become attached to the mitotic spindle via kinetochore microtubules that attach to the kinetochores, which are bound to the centromeres. As prometaphase is nearing its end, the sister chromatids are observed to jerk back and forth between the two poles. During metaphase, the sister chromatids become localized in a region called the metaphase plate. Once the chromosomes have aligned at the metaphase plate, metaphase is complete. When metaphase is completed, anaphase begins. The sister chromatids separate, and each chromatid is now linked to only one pole via a kinetochore microtubule. The kinetochore microtubules shorten, and the individual chromatids, now called chromosomes, are drawn to the poles. Toward the end of anaphase, the polar microtubules push against each other and cause the two poles to be moved farther away from each other. Each daughter cell will receive the same complement of chromosomes as was originally found in the mother cell, thus assuring that each daughter cell gets an identical copy of genetic information. During telophase, the chromosomes decondense and most of the microtubules depolymerize. Portions of nuclear membrane surround each individual chromosome, creating vesicles. The vesicles fuse with each other to create a nucleus containing all of the chromosomes at one pole of the cell. In animal cells, cytokinesis or cell division involves the formation of a cleavage furrow. The plasma membrane is observed to constrict and eventually separates the cell into two daughter cells. The daughter cells then enter interphase, where growth and enlargement occurs, leading to either preparation for another mitosis or differentiation and specialization occurs.
In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells.